All right, today we are diving deep, and I mean deep, into the genetic code of our food. We're going to talk about a tool that is absolutely changing the game for agriculture. It's called SNP.
You know, this is really the central question, isn't it? We've got more people to feed and a climate that's throwing curveballs left and right. We need crops that can take the heat, fight off new pests, and give us bigger harvests. And we need them like yesterday. The answer, as it turns out, is hidden in the blueprint of life itself.
Okay, so let's get into it. To really get why this new tool is such a big deal, you have to understand the hurdles that crop breeders have been up against for, well, forever.
For most of history, right? Crop breeding was just a slow, painstaking game of observation. You'd cross two plants and then wait and hope for the best. Then, the 80s came along and we got our first peek at the DNA level with things called RFLP markers. They were revolutionary, but man, were they clunky and expensive. The 90s gave us SSR markers, a huge step up for sure. They were faster, more reliable, and became the go-to for a while. But they still weren't quite enough for the massive scale we needed. The stage was set for something totally new. And that's where our hero comes in, the SNP marker. So what on earth is an SNP? Okay, the full name is a mouthful, single nucleotide polymorphism. We just call them SNPs, pronounced SNPs. And a SNP is basically the most common type of genetic variation you can find. I want you to imagine a plant's entire genetic code, its genome, as a massive instruction manual that's millions and millions of letters long. A SNP is just a one-letter typo. And this picture just says it all, doesn't it? That one single tiny change, that one-letter difference in the entire blueprint can be the reason one plant gets a disease while its neighbor stays perfectly healthy. It can be the difference between a crop that can handle a drought and one that just gives up. It's a tiny, tiny clue that can have massive consequences. So, here's why SNPs are such a game changer. Think about it like this. The older markers were like having a map with landmarks every 10 miles. With SNPs, it's like having a signpost at every single street corner. They are, by far, the most abundant type of genetic variation, which gives us this incredibly high-resolution map of the genome. And the best part? The whole process can be automated, which means we can analyze way more data, way faster, and for a lot less money. It's a total win-win-win.
So these little markers are everywhere, and they're super powerful. But how in the world do scientists actually find these single-letter typos among billions and billions of letters of DNA?
Well, it's a super cool three-step dance between biology and computer science. First, you use these incredible machines for what's called next-generation sequencing, or NGS, to just read the DNA from tons of different plants all at once. Step 2, you feed all that data to a powerful computer, which lines up all those DNA sequences either against a master reference copy or against each other. And finally, the software just scans through and flags every single spot where there's a one-letter difference. Boom, that's your SNP.
And the scale here is just mind-boggling. We're not talking about finding a few hundred of these markers, or even a few thousand. With these new technologies, we're finding millions of them in our most important crops. It is an absolute data explosion.
Okay, so once you've found the SNPs, the next job is genotyping, which is just a fancy way of saying you check which version of that one-letter typo a specific plant has. And scientists have a whole toolbox for this. If they only care about a few key SNPs, they can use some very precise, small-scale tools. But if they want to see the whole picture, they use these massive platforms that can read thousands or even millions of SNPs all at the same time.
All right, let's get to the really exciting part. This is where the rubber meets the road. How is all this amazing science actually changing the food we grow and eat?
The impact is huge, and it's happening across the board. We are literally accelerating how fast we can develop new crops. We're talking higher yields, better nutrition, crops that can handle heat and drought, and, this is a big one, crops with built-in resistance to all sorts of nasty diseases and pests.
So, let's take maize, for example. Using SNPs, scientists have been able to pick apart really complex traits like exactly when a plant flowers. They found the specific genes that fight off major diseases like northern leaf blight, and they've even pinpointed the genetic location that leads to higher oil content in the seeds. This lets them select the best plants when they're just tiny seedlings, which speeds up the whole breeding process like you wouldn't believe. And it is the exact same story in wheat, a crop that feeds a huge portion of the world. SNPs were absolutely critical for mapping the gene that fights off the devastating Fusarium head blight. Breeders now use them to screen for resistance to all kinds of rusts and mildews, and even to select for genes that control things like grain hardness or plant height, which are both super important for getting a good harvest.
And one more, just to really drive it home, soybeans. SNP markers have let breeders just zero in on the genes that protect soybeans from their biggest enemies. We're talking about resistance to aphids, to destructive nematodes in the soil, and to viruses. The point is, this isn't science fiction. This is happening in labs and in fields right now.
So when you step back and look at the big picture, this technology has just completely rewritten the rules. It's given us a high-definition roadmap to all the incredible genetic potential that's been hiding inside our crops all along. This quote from the research really just nails it. Before SNPs, our genetic maps were, well, they were pretty sparse. Now they're saturated. They're so full of these little markers that scientists can track almost any gene they want, zoom in on a specific region with pinpoint accuracy, and dramatically speed up the process of finding and using the genes that really matter.
And that, of course, brings us to this final, really fascinating question. For pretty much the first time in human history, we can read the instruction manual for life with this incredible speed and precision. The power to design crops that are more resilient, more nutritious, more productive. It's right there in our hands. The question isn't really, can we do it anymore? The real question is, what do we choose to build?